So I'm so excited to sit down today and talk with Dr. Charlotte Deane. She's a professor in the Department of Statistics at the University of Oxford. And last year in 2025, she was elected as a fellow of the International Society for Computational Biology. Um, Charlotte leads the Oxford Protein Informatics Group, who work on diverse problems across immunoinformatics, protein structure, small molecule drug discovery, using statistics and AI to generate biological and medical insight. So in our community, what she's best known for are the novel algorithms, tools, and databases, which are openly available to our community. It's hard to imagine anyone doing antibody drug discovery right now who's not using SAbDab or aBodybuilder or Therapeutic Antibody Profiler. And for those of us working in single domain antibodies, we're extremely grateful that there are nanobody specific versions of all of these resources. So kind of my first question to you, Charlotte, is when did you first start working in nanobodies and how did you get interested in them?
So I suppose it's a double answer. I first started working in antibodies almost exactly the same time that I first really started working in antibodies. So right back at the beginning when we built SAbDab, so we published SAbDab, I think the original version in 2014, and we were working on it before then, and we just because we needed the data, we'd made the database, and then we were like, well, we'll make it available. So it was a bit before that where we eventually. But even within that, we were collecting the nanobodies because they're just a form of an antibody. So they were collected then. But it was really as we got into sort of 20, I guess, sort of 2020, 2021, where we started doing a lot more work where we were directly looking at nanobodies and tried to make nanobody specific versions of a lot of the things we were thinking about. But all the way through, it was kind of there because when we built our original observed antibody space, we collected VHH sequences even at that time. And there are VHHs in the database of our antibody sequences because they're just another form of that. So it's kind of been a sort of mixture between the two. It's always been there. And then we've picked up the pace now because there is enough data to be able to make much better tools off those things that are specific for nanobodies.
Yeah. So you said that, so SAbDab, you started it in 2014 or 2015, I think you said?
Yeah.
So at that point, there were nanobodies, but they weren't a huge fraction of the structures that were in the database, but that's rapidly increasing. And it's kind of, I guess, do you see it as a leading indicator since I think it's one in five structures in the database are nanobodies now, but that's certainly way more than there are clinical nanobodies kind of
Yeah.
in kind of approved at this point. So what do you think is
So
driving that interest?
So I think there's a whole set of things that are driving that. So I think you could jump immediately to going, well, there has been a definite increase in the therapeutic interest in nanobodies and the diagnostic interest because we started to see, there are actual medicines on the market which are nanobodies, and you can see them developing through, and there's been a lot of interest. But because what we're collecting there are structures, there's another effect which is allowing us to get more data, which is probably also helping us on the therapeutic side, is nanobodies have been seen as a really good tool molecule because they're small, they're rigid, they tend to be very site specific, make them with high affinity. So they're great for stabilizing X-ray crystallography for structures and membrane proteins. They're fantastic as sort of cryo-EM chaperones. So they can increase the particle size, they can improve, they do really good stuff. And because cryo-EM has become such a big technique, we'll quite often have lots of structures where we've got different nanobodies even to the same protein where they've been trying to get different viewpoints or structures of a given protein. So that gives me a lot more structural data around them. I think the other thing beyond that is that it's always been true, but it's almost like people only really picked up on it in the last sort of 5, 6 years, is nanobodies are really easy to generate at scale. So antibodies are easy to generate at scale, but nanobodies are a lot easier because they're a single domain, so that's much simpler. They're easy to express in bacteria and yeast, and they are very stable. So that gives them loads of advantages in terms of just making them and generating them and their use as a tool molecule. And then sitting alongside that, because that started happening, I think we've got this increasing therapeutic interest because we can see more and more what they can do, and we're starting to believe that they would be in some cases, the better form of therapeutic you could make for particular targets or diseases.
Yeah. So it's kind of, I guess, partially interest driven, partially a happy accident of them being just used in a lot of structural data to begin with.
Yeah.
Oh, interesting. Okay. So kind of, I guess, off of nanobodies a little bit, one of the papers that you wrote that I thought was kind of most interesting had nothing to do with antibodies. It was called 10 Rules for a Successful Cross-Discipline Collaboration. And I thought that one was kind of particularly interesting because one of the rules that stuck out was go to the wet lab. And that was, as a computer scientist, I can see that that is extremely hard actually for people in computer science to do. And I was just curious about what your kind of how you arrived at that as being kind of in the top 10 rules.
So I wrote that paper some time ago, even sort of before we had the machine learning explosion, but I hold by almost everything we wrote in it because the basic principle is that we always think that the job that the other person is doing in a collaboration is somehow easy and doable. We kind of know it's not, but we don't understand it properly. And that's the reason you're collaborating because, I collaborate with experimentalists because I'm quite good and my group is quite good at generating code, building databases, computationally making predictions. But I was appalling in a wet lab when I worked in a wet lab a very long time ago. It was only time I've done it was when I was actually an undergrad. So, it's a very long time since I've been in a wet lab. But that it's really important to try as far as possible to understand what your collaborator or co-workers are attempting to do and understand how hard it is to get the data and where you can and can't get the data. Because one of the things that happens often as a computational person is you say, well, can't you just make 1,000 of these? Because I could answer the question. And sometimes you can, because sometimes you can just do a library and you can just screen it. But if I say, I really need 1,000 precise binding affinities right now, yep, that's a really big deal. Yep. And unless you understand that, that's very hard. And then the other part of it is it's like the time it takes to get the data, the error that might be in the data, the way the data works. And also because the language that we use is different. And the easiest way to learn someone's language is to be in their space and not in yours. So when I say the word model, I have a very particular meaning of it in terms of a computational model that does something. If you say the word model in a sort of normal human setting, nobody is thinking that. Yep. They might be thinking of a very good looking person who walks down a catwalk. They might be thinking of a car kit, depending on who you're talking to. And you have the same with lots of language in science is like that. And it's very difficult to find where those issues are until you really spend some time truly understanding what they're doing. That doesn't mean become an expert, but it's important. I think it's really important. And I often think that lots of the really big mistakes that come out in computational papers come out because the computational team doesn't understand the experimental data they are dealing with. And it's not stupidity, it's literally just a lack of that connection in that way.
Mhm. Do you prioritize that in your group? Do you encourage your students
Yeah.
to do both? Yeah.
So I encourage my students not necessarily to do work in both, but to have enough time that they will go and visit the labs that are producing data they might work with or spend time with. I think it's very hard to become really deeply expert at running really good wet lab experiments because it is actually really hard, as well as being really good at writing code. Some people can do it, but more commonly what you want to be is I'm really, really good at writing. I love working on a computer, writing code, but I should make time to make sure that I can spend some time in a wet lab, even if it's only a week. A week is long enough to get a really good feeling. I need to talk to these people properly and understand and help and be as useful as I can. And
Hmm.
so I think that's the important thing to try and get into people's heads. So it's making sure there's time for that. And also, if you can't do that, spend a lot of time talking to people who do work in the wet lab. Don't just go take the data off a website and run away with it. If you don't understand the data, you must talk to the people who generated the data in some way,
Yeah.
because otherwise you'll interpret it incorrectly.
Yeah. And hopefully the people that generated the data had some thought also to talk to a statistician or a
Yes.
informatics person beforehand, right, when they designed the experiment. So both ways.
Yes, it's always both ways, but I tend to have to appeal to my community to behave well, and then others can appeal to theirs in the other direction as it goes.
Right. So then kind of our audience is mostly wet lab scientists who are developing therapeutic antibodies. They're in kind of the discovery phase. And I think for them, it's been sort of the last few years, it's been a little bit overwhelming in terms of what computational tools are available. People talk a lot about, there is hype. And so I think they wonder what's real, what's not, how do I get started, what tools are kind of ready for prime time, and what tools are going to lead me down a difficult path. I guess, do you have any advice for those scientists about how they might get started, kind of
Yeah. It's
understanding what's going on?
To be fair, I think it's overwhelming even if you're a computational person right now. The speed and volume which new things are arriving, the ways where it's very hard to tell what's been validated, what hasn't, what's hype and what's not. And that's not just limited to the antibody field. That's kind of across the board as a computational researcher, you can see this, but you see it a lot in the biomedicine, and you can see it particularly around antibody design with this. So coming from the experimental side, I think the best way to think about it is think about the process that you are doing as an experimentalist that you would do. You think about some form of design of what you're thinking about, some form of screening, some form of optimizing, thinking about de-risking and then selecting and then repeating that process, usually going around in circles until you get something you're happy with. And then in terms of the tools, the skill is to get some knowledge of what they're already good at. So I can give you things that the tools are good at right now. And I'm not going to say it doesn't matter at all which tool you pick up, but it matters much less in these areas because actually a lot of them are good at it. So one that's true is structure prediction. Now you could run straight to AlphaFold for that. I'm obviously going to say if you're doing nanobodies, use Nanobody Builder or a bodybuilder, so the things that have come from my group, which are about as accurate as things like AlphaFold, but an awful lot faster. So it makes it easier to process a lot of data. Now, those tools are out there, and structure prediction, we're good at. Computers can do that. So single structure prediction of your thing, you can do. Other things that are there quite a lot are ability to do developability risks. So this isn't perfect, but computational, assessment of developability. So I'd go down the route thinking about my tools. So therapeutic antibody profiler, therapeutic nanobody profiler that now exists. So you can do it like that. But also, there are other tools, things like CAMSOL or Aggregate, where they're trying to test the aggregation. And actually, those tools are not perfect, but they're really quite good at giving you an idea of the space you're operating in and at least telling you the really bad stuff you've got. If you've got a sequence, they're good at that. And then the other things they're good at, things around library design, being able to use the language models that exist. We can do library design, we can design better libraries. And then things that are sort of actually surprisingly we are very good at. If you already have an antibody or nanobody that binds, improving its affinity. Yeah, not every single, I can't write down exactly what will give you the ideal affinity, but there are a lot of tools out there designed around the concept of doing that and rethreading sequences onto it. And we can do that. And we can do things like improve their stability. Yeah. So once again, it turns out actually sequence changes using large language models, very powerful for doing those kinds of toys. So thinking, from the kinds of things we have, things like ablang for doing that kind of thing, but you have other antibody language models that have been built in various shapes and forms of different types. And those models also are the things that are driving humanization tools. So we have humanization tools that will help people try and do this, but so do lots of other people. And in general, you can potentially improve the humanness of your antibody or nanobody. It's hard with nanobodies because we don't know what a human nanobody looks like because it's not a natural thing in the system. So I always struggle slightly there. But with an antibody, you can use things like Humatch and Humab and that kind of thing to do that. Things that they're very bad at are absolute affinity prediction. Can't do it, right? So relative affinity prediction maybe. So if I know how well this one binds, I might be able to tell you if this one goes up or down, which is what I was saying maybe good at. Absolute, how good it really is, no, on average you won't get. Real immunogenicity, not a chance. Yeah, we just don't have enough data to do that. So there's a lot of proxies between us and that. And then there's the kind of in-between bit. So I could go other things they're bad at, but they're kind of obvious, like PK/PD and in vivo efficacy, where, yeah, those exist, but those are not things that computers can do yet. And then you have the in-between bit where I think this is where people are struggling to get an idea, which is, can these models predict me the complex of my nanobody and my antigen? If I know this is my nanobody and I know this is my antigen, will it get it right? And the answer to that question is sometimes. And that's where it gets hard because it depends on exactly what your nanobody looks like, what your antigen looks like, how much information you've got, how much it looks like something else that's been seen in the data sets, whether using something like AlphaFold Multimore or AlphaFold 3 will actually correctly fold these two things into their correct binding pose. So that's where I give that's the wishy-washy answer, where I say I would run the tools, but as with all of this, I wouldn't trust them completely. I'd use them as hypotheses generators for yourself within that. So that's a kind of how I would describe the landscape. Does that give a kind of feeling? So the place to start is to start with, if I wanted to just play, I'd play by doing structure prediction because those tools work. You can see it, you'll get some things that are useful. And then I do things like developability prediction on that, see if I can do residue changes that will improve affinity because those are all quite standard. And then I'd move towards thinking about whether I can do things like co-folding, docking, those kinds of things.
Hmm. Is it just a data problem? So you mentioned kind of like I think the therapeutic antibody profiler is kind of a surrogate for some early kind of antibody screens. And those are kind
Yeah.
of experiments that maybe we have a lot of data for because it's kind of like fast and early. But all the PK stuff, that's kind of like late stage, right? And we don't have as much data. Is it just a data problem or is there something more
Yes,
complicated that we can't model?
I think fundamentally it's mostly it's quite hard because in one sense I think fundamentally it is a data problem. The problem is that that data problem might be so big that it isn't a data problem because if you needed 1 billion data points, you're never going to have them. Therefore, it isn't just a data problem. And then so that there is a data problem where you have more data, the whole kind of machine learning explosion works much better. Where you have small amounts of data, you have to think of methodologies which aren't based on machine learning. So actually, the therapeutic antibody profiler is not based on machine learning because we have, several hundred antibodies that have made it to phase 2 clinical trials. So that's enough to do kind of standard statistical work with, which is how the therapeutic antibody profile works. It's not enough to write a large machine learning model with. When I come back to something like humanness, yeah, OAS has 2.5 billion sequences in it. So I've got 2.5 billion sequences of single chains of antibodies. I've got about 2 million odd non-redundant antibody paired sequences. So I can build large language models there. So I can do humanness prediction with a lot of data and I can use a machine learning model. So it isn't quite just data because it's how the data is distributed, how much you've got, the types of ways you can feed it in. But if you're going down the machine learning route, it is basically just data, yes.
Hmm. I guess then also kind of, so as a computer scientist, if I'm developing a tool, I'm maybe checking against maybe tens or hundreds of labeled data sets to say how accurate my tool is at calling the right thing. And so for me, maybe 90% accuracy or 95% accuracy is good enough. But for the experimentalists, they're kind of focused on one target. And so what I think, would be helpful for them would be to understand where are the biases of the model where that 5%, because I'm working on ion channels or I'm working on something that's, not homologous to anything known before, or a glycan or something that really isn't kind of well understood. I guess how easy is it for them to assess what the bias of the model is that it would work for their one campaign versus kind of like broadly accurate?
So, I mean, the first thing to say is that humans aren't 100% accurate when we decide what we're going to do next
Yeah.
with the molecule. So, I always think it's important because everyone's like, well, the computer's only 90% accurate. I'm like, have you ever got it right 100% of the time? I certainly haven't. Not in anything I do. But so the point here, I think that one of the issues with the moving into the world of machine learning is that it's very hard to properly have an estimate of the likelihood of the accuracy of your outcome. It's not impossible, but it's very, very difficult because there isn't an underpinning statistical model with these. And because there isn't an underpinning statistical model, I can only work out how accurate it is by doing those kind of benchmarks you talked about. I just do lots of benchmarks and I say, well, 90% of the time it gets it right. And if I do enough different types of benchmarks, I can say, well, in these cases, it's more likely to get it right. And in these cases, less likely kind of thing. So I can give you errors, estimates and that kind of thing. But when presented with a piece of data, so a case that comes from outside its training data, the behavior of the model is, in some sense, we don't know exactly what it will do. So it might get it right, but it might get it wrong. So it's always best to use all of these tools the same way we would use advice from another human, which is, okay, it's pretty accurate, should be useful. Think about it. Does it make sense? Would I do it? And the more your target you are interested in or the thing you're working on is homologous or similar to known other data, the more likely it is to be correct. There's lots of papers that show this. But just because it's very different doesn't mean it will definitely be wrong. So it's still worth looking at those answers and then thinking about it, because before we had the tool, you wouldn't have had anything to look at. And so you would have just been, in a sense, making it up with your own brain. Here, this is another input to help you with that. And you can do it multiple times and have multiple different inputs. But none of these models are reaching 100% accuracy at the moment, none of them. And lots of them are useful if you can take 20, 30 answers out of them and test all of them, or look at the top 50 and pick 7 or 8 that look sensible. So it's always worth thinking of them in that way, and that's the best way to think about them at the moment.
Hmm. Yeah, I think people would be happy if they could look at, like, if they could get 10 to 20 out of something. I think what they're worried about is if it gives them 10,000 predictions, how could they possibly sort
Yeah.
through all of that?
Yeah, and I think one of the skills there is to remember you can always tell the model not to do that, or you can tell it you only want the top 10. Because as a computational person, I quite frequently might want to make my model generate thousands of things, because I might then be computationally testing it against something else. Yeah. But all of the models you can control and just go, unless you actually are going to test it computationally as your next phase. Because
Hmm.
yeah, if you're a human, if you go above about 50, you lose all sense of interest in any of it.
Hmm. Okay, so then in terms of the kind of the questions that our customers ask about what can, software do for me today, you mentioned optimizing a binder for affinity is doable. So
Yeah,
kind of I have an antibody, I have a target, I can make it bind tighter.
you should be able to make it bind tighter. Now, it's not that the computer will get every residue change right, but if you use those kinds of tools and ask it to make predictions, you will get a relatively high hit rate of the changes it suggests will improve your binding affinity. The
Hmm.
thing that you have to watch for is being clear about how you would check the specificity of your antibody and nanobody as well. So in general, it shouldn't cause a problem there, but it's not. Most of the methods are only optimizing against one function. So they optimize increasing affinity, which may or may not make it more specific. And that's just to remember, all the other dimensions of your optimization as you do these things.
Yeah. So then the next step that a lot of our customers need to take is humanization. And I guess the humanization you mentioned, kind of like making it look human, is possible because we have good models for what a human heavy chain antibody should look like.
Yeah.
But then retaining affinity while we're humanizing, is that something that it can think about both at the same time?
Yes. So you have some models that think about both at the same time. I mean, often the way we do it is we don't necessarily think about both at the same time. What we do is we tell the model to not change any of the parts that we think are really important for affinity. So don't change the CDRs, don't change, you just say, don't make any mutations where I don't want you to. That's a very simple way of doing it. And that tends to be the best way to start because it's very simple for the model to optimize under those conditions. And the other thing is, of course, you can do this as an iterative process. So one of the things we would do is, I think this is quite an affine antibody, but I want to humanize it. I run the humanization code and then I go back and I build another model of it. Does it look identical to the model of because structurally these two things should look exactly the same if it's going to bind exactly the same way. So if anything I have done has upset that structural shape, then that was a bad choice. So you can kind of do this yourself, but there are models now starting to come out where you optimize multiple things at once within a single model. But sometimes I still recommend doing things like, because structure prediction is so cheap and so easy now that constantly, if you get a new sequence, predict the structure and it should look the same as a prediction or the crystal structure of the thing you care about. And if it doesn't, you should worry because even if it does, it might not be the same. Yeah, you can't prove that. But if it doesn't, that means the model really thinks something has happened because they're fairly similar sequences. So they should
Hmm.
come out the same. On average, they should come out the same. So if they don't, the model is saying there's something happened here. And so you should worry about that and go back round and be able to check.
Yeah. And then you mentioned that absolute affinity prediction is not necessarily possible at this moment. So I'm guessing the kind of corollary to that is predicting like an epitope is not necessarily possible, or conditioned on that it is a binder, you could predict the epitope.
So you have various different versions of that, which all depend how much information you have. So there's I just have a sequence of let's say I have a sequence of an antibody and I have a sequence of my antigen, and I know they bind to one another. So you can run co-folding software or AlphaFold multimer and try and get a complex, and that will sometimes give you the right result. Yeah, but the hit rate is really does depend on whether it's seen something relatively similar in the data sets before, and its actual hit rate on things it hasn't seen before can be quite low, like 20%. So that might not find you the epitope that you're binding to. But if you have any other information around this, you can start to get loads better. So that's the, if I know nothing, I just know these two bind. So for example, one of the things we've shown, we have a tool called Space, but other people do similar things where you can show if I have, so for example, for COVID, it's a great example to work on. I've got tons of data. You give me a new antibody for COVID, yeah. I could just compare its sequence to everything I've got already. And if it's similar to one of those, it probably binds in the same place. So if the CDRs are similar enough, yeah. You can also now just predict its structure, predict the structure of all your other binders and check that its structure is similar. And if structure and sequence are similar, it binds in the same place. So you can already, that's it. Then you know the epitope because you knew the epitope of the other one. If you have any information to constrain by, so you happen to know that this residue is definitely within the epitope, don't know where it is, you just happen to know that that's probably part of it, or you've got another antibody that binds where you do know the epitope and they compete or they don't. All of those things can be added in as constraints or restraints to these models, and suddenly you'll get the chances of getting the right answer just go up really quickly. You know, you need tiny amounts of information to massively improve the performance of these models.
So when you said, okay, so when you said that knowing the antibody and knowing the target is knowing nothing. And I guess what happens if you know less than nothing? So you have, let's say you immunized a llama and you immunized it and you know what you immunized it with, and now you're looking at the repertoire. Could you basically predict that some of these bind or don't bind? You have a little bit less than nothing information, but not really nothing information because you know it's been hyper immunized.
Yeah, so then that comes back to the use of kind of an examination of what's coming out of the repertoire. So you're looking for the things which are, groups or families of antibodies that are really overexpressed together, where there's lots of them. And once again, because we've got very fast structure prediction tools, you can do the way people would do currently, which would be basically do clonotyping and look for clones where you've got lots and lots and lots of that clone. But you can, alongside that, you can do structure prediction. So you can look for the same structure coming up again and again and again. So you can then have things. So this is something we found a lot, which is the sequence. So clonotyping works, right? Things where if you immunize and then one clone you see tons of, it's very likely that's a binder and there's, that set of sequences is a binder. The interesting thing is that you can see more diversity in your binding if you look at the structural overlaps, because you can look for all the structures and then find all the structures that are the same. Yep, and they might come from different original V genes, but they've taken up the same shape. You can see that has now become a very large grouping of the similar structure occurring a lot. You can't necessarily see this in sequence space, and that's another indicator. So you can start pulling those out as the kind of binders, as the sort of standard way you pull out for an immunization library.
Mm-hmm. So kind of organizing your repertoire based on sequence is one way. Organizing your repertoire on structure is kind of another way to help prioritize these clones.
Prioritize. And when you do this, the absolute best bits of information is when you have something where you have lots and lots of similar sequences which also share similar structures, because you can have things which are the same clonotype, which you would predict to have different structures, because clonotyping is just a measure of, sequence diversity, and particular residue changes can cause structural change. So what you want is a set of sequences which are all very sequence similar and structurally similar. And that is, you can make quite big bets then.
Mm-hmm. I guess one problem that people often have is they want to make a nanobody that functions the same as a conventional antibody they have. Maybe they have a mouse antibody and they want to make a nanobody. Is it possible to single chainify the conventional in any way other than making it an scFv?
So you mean as in actually generate an antibody that binds to that epitope? I mean, people are starting to build methodologies that do that, where what you do is you take your antigen and this part is the epitope and you know exactly what residues are involved in the binding. And then you try and literally put an antibody there and build loops. So you're kind of in-painting a structural thing that will bind there. Those methods have shown different levels of success, particularly so if you go from antibody to antibody. So it's almost like if you took an antibody and ripped off all its CDRs, but held it in exactly that place across over the antigen and then said, right, rebuild my CDRs, trying to fit them against that. Um, that does show some form of success, not
Mm.
always, but you can get some results which are really good. And there are things that run through things like RF diffusion and methods like that, which will allow you to do this kind of design, and you can go through protein MPNN and you can build it. But it doesn't work all the time, but it does. Changing the actual backbone causes you a problem because it's like the change from one to the other. I think those are the kinds of tools that are coming now. So I think people are doing that kind of thing, but as an off-the-shelf tool or a way of doing it is not there yet. But I think it's the kind of thing that's starting as I suspect if you ask me this question in a year's time, I'd be pointing you at packages that would help you do that, but I can't do that today.
Okay. All right, 12 months. We heard it here.
Yeah. I reckon it won't be that long because it's a way people are thinking about this problem, but it's not trivial. So it may turn out that you just can't do, as in it won't work very well because there's not enough data, but it's a way people are thinking about trying to do these things.
I guess how close do you think we are to not having a lab in the loop or not having an immunization in the loop for this?
I think this is really hard because I think there are very differing opinions in the literature about how close we are to this. I think we are still a ways away if it is a target that we have never done any work on before, so something that's actually novel and new. At the other end, if it's something, so if we're working on the receptor binding domain, I'm going to pick COVID again because everybody
Yeah.
knows there's loads of data. But if you pick the receptor binding domain of COVID and you took all the data we had, trained something, you can do really kind of quite cool things. So we did work which showed that we could move it from binding to one receptor binding domain to a different version of the virus. You could give us an antibody that bound to one. We could move it across to bind to another. We'd only have to do, say, 6 experiments or 12 experiments, and we get 50% hit rate, 75% hit rate. So I'm only designing 6 or 12 sequences. I could do this, and I could improve its expression within that. And you can do that. But that's because there's so much data there that I have a really good understanding of what the antibodies look like, even if I don't have any data about exactly the new proteins, the new antigen, because it's got a few mutations in it. I have such a good understanding of antibodies that bind the receptor binding domain. It's so much data. Yep, the models are actually quite good. Yep, they just are. So, but at the other end of the spectrum, when it's brand new, it's much harder. So there are more and more papers coming out suggesting we are getting better and better at this. I have a suspicion that we're still a very long way away from what I would call it being generalizable, as in if it's a very different target, that's just so hard to imagine these models getting that. But the amount of experimentation we will need will go down, is what I'm hoping. So as soon as you have a small amount of data, the model will just kick in and allow you to do far fewer experiments. So in the example I gave, instead of having to do thousands of panning screens, yep, here are 6 sequences you need to make, and 4 of them work. Yep. That is incredible, but it didn't rule out experiments because it wasn't all 6 worked. And in some cases, we did 12 and only 4 worked. So that's, you have to do the experiments. So what I think is going to happen is the number and volume of experiments we need to do drops, and the types of experiments that become important, will drop. So we will be able to say, do all of this computation, and then you only have to do this experiment. And that will tell you enough that you can move on to the next stage.
Yeah. I'm sort of hoping, I mean, so COVID was sort of I don't know that that would ever happen again for any particular target that we would generate that much data. But the technology is allowing us, like cell-free expression, like you said, cryo-EM, everything is becoming higher throughput and faster and cheaper. And so I'm wondering if it doesn't need a global pandemic and we could still generate something of that magnitude for like more targets, if that isn't becoming more possible experimentally, and then that kind of gets us to where we need to be for the software.
Yeah, so I mean, in my sort of other world where I'm working with small molecules, I'm involved in a project called OpenBind, where our entire purpose, I mean, I'm not doing the data generation, I'm working on the computational side, but the purpose is to try and generate as much data as possible, to be able to get a more generalizable model of protein ligand binding. And we can do that now because you can do high throughput crystallography is what we're doing, and then high throughput binding affinity experiments. Yep. And it's non-trivial because you've got to, have enough protein, you've got to have the small molecules you can use. I actually think in the biologics world, it's even easier. The way the experiments are set, the way because you can just make antibody libraries or nanobody libraries. As I said right back at the beginning, one of the reasons everyone's using nanobodies is it's actually really easy to express them. They're stable. You can make big, you can do this. And the experimental setups to check if they just bind to something are relatively straightforward. And the cryo setups are becoming easier and easier for us to actually get even very basic structure. We don't need detailed structures, basic structures, because once I've got a basic structure, something like AlphaFold will make you a really good structure. Yeah. So part of me is like in the biospace, it should be even easier to do what you're arguing for. The problem is I've spent a lot of my career asking people or saying to people, please, 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 you should make more data. You should make this kind of data. It'd be really helpful if everyone released their data, and discovering that it hasn't happened yet. So I'm still a believer, but I think we've got a ways to go yet.
Yeah. So for us, llamas and alpacas kind of dominate our thinking and the kind of nanobodies that we look at, but are there other single domain antibody modalities that are kind of interesting that you've looked at?
So there's one other, we've looked at both of those. We have looked at some others. My favorite one, which is shark VNARs. They're really interesting because they're actually a little bit different from all the others because of the loop they're missing in their construction. They're really interesting computationally to try and number and try and find, but they also look like they have some interesting binding properties as well. And the final part of this is the obvious thing, because if shark VNRs turned out to be really important, then I'd be allowed to have a shark tank, obviously.
Yeah.
So why would you not pick them? I mean, llamas are cute, but as shark tank.
Yeah. Have you looked at all at the cow ultra long CDR3s? Because a lot of people have talked about those as being kind of single domain-ish. I mean, not
Yeah,
single domain in the sense that they look like antibodies, but a small binding fragment
they're a
derived
small
from an
bite.
antibody.
I've done tiny amounts of work with those, and they're quite interesting to try and model because of the way they're basically held together by disulfide bonds and things like that, which has other implications if you're going to use them further on down a pipeline as a therapeutic, that you've got this sort of dependency on those. Um, they are surprisingly hard to do structure prediction of because there's
Oh,
very little data around them. And the disulfide bonding piece makes it interesting and should make it easier. But I think they are another interesting category. I've only played with them a tiny bit.
great. Well, I think that is all my questions. Is there anything else you wanted to bring up?
No, I think the only thing I'd say is if there are lots of biologists listening or people who work in the wet lab and are starting to play with the thing I'd really say is go and play with the computational tools. Yep. Because lots of them are available as websites now or things like that. And even if you can't put your own sequence in because it's proprietary, go and play. Because the difference between a computational tool and wet lab work, the real difference is, it's one of the reasons I can do this and I can't do wet lab work, is you can't really break it. Yeah.
Mm. That's a challenge.
Literally. Well, but you can crash the computer, but it's not the same as the effects you can have in a wet lab if you are as clumsy as I am. So it's the best way to learn and get a feeling for this is find some of the tools where there's a website or something where you can just put a sequence in, put the sequence in, get it to produce the structure, put the sequence in, see what it shows you about developability, so that you can see how they might be useful to you. And then once you have a little bit of a feeling, go and talk to people who are using them. Just go and ask them questions, because let's be honest, most people love, just like I'm doing now, love talking about what they do, and they love talking about their science. So they'll help you without a doubt.
That's great. Well, I appreciate it. Thank you so much. And I hope you have a wonderful weekend.
Thank you.